Okay, so this is our week 5 lecture for microbiology, general microbiology. And we are going to be focusing on microbe and human interactions, contact infections, and disease. Right, so there's a lot of lots of new terminology in this unit. So that's essentially what we're looking at over here. So if you look at host, right? What's a host? Any organism that harbors another organism, or a particle such as a virus or a viron would be classified as a host. Now, what's symbiosis? So symbiosis is, we can look at that as an association between two species in which they live together. And often this is in very close physical proximity, and they have an impact on one another's survival and as well as each other's reproduction. Mutualism it's a type of relationship between two species in which they both benefit from one another in their presence. So, for example, E. coli bacteria that live in our large intestines, they end up producing vitamin K, right, which is good for us. And they have a place to live in, and they get food from us that we eat. So again, we both benefit. Parasitism. Parasitism, this is a type of relationship between two species in which one species benefits at the expense of the other. So for example, you can look at a tick. It feeds on the blood of a host. It causes harm to the host, while the tick benefits itself. Commensalism. So this is a type of relationship between two species in which one species benefits while the other is neither helped nor is it harmed. So for example, barnacles that attach themselves to a whale. The barnacle gets benefited. Why? Because it's being transported to different parts. Wherever the whale is going, that's where it's being taken to. Now, the whale gets no benefit from him, nor is it getting harmed by the barnacles. Okay, so that's commensalism. Right, now, most microorganisms are harmless and they don't cause any harm to humans. However, a few microorganisms, they can actually benefit human health, while even fewer can directly cause harm. The normal microbial flora, okay, also sometimes it's called a microflora, or sometimes it's called a microbiome. When you see these terms, it's referring to the microorganisms that are usually found in association with human body tissue. Okay, this would be the microflora or microbiome. And now, these microorganisms, they can be helpful. For example, they could be helpful in aiding digestion and providing protection against harmful bacteria or virus or even fungi. Resident microflora refers to the microorganisms that are always present on or in the body, while transient microflora is referring to microorganisms that are present for short periods of time. And again, it could be maybe a few minutes, or it could be months. Now, the adult human body is made up of about, look at this number, it's huge, 10 trillion eukaryotic cells, right? Again, which are human cells. And we also have a staggering, look at this, 100 trillion prokaryotic cells, right? And these are the bacterial cells, right? So in our body, we have 10 times more bacterial cells than human cells. It's, try to grip on that. So, resident microflora can be found in various areas of the body, such as your skin, mouth, GI tract, as well as the reproductive system. So, the absence of resident microflora can have harmful effects on the body. Why? Because again, as we said, that some of these microorganisms, they are helpful, right? So they play important roles in digestion, in our immune system function, and also in protection against harmful bacteria. So they've done studies on cattle, and they found that the absence of resident microflora can actually lead to harmful effects, such as in cattle, that is, enlargement. They have an enlarged cecum. They also find vitamin deficiencies, as well as underdeveloped immune systems. Microbial antagonism refers to the ability of the normal biota in the body to prevent the colonization of harmful microorganisms by competing for attachment sites and creating a hostile environment. So there's a limited number of attachment sites in the body, which limits the number of microorganisms that can actually colonize a specific area. In addition to that, the chemical or physiological environment that's created by the resident biota can be hostile to other microorganisms. This is what's going to prevent their colonization. So the normal biota in the body is usually beneficial, and at worst, it's commensal to the host. So this means that they don't cause harm to a healthy host, and they instead, they may even provide benefits such as aiding digestion and again, protecting against harmful bacteria. All right, first acquiring resident microflora. So the first acquisition of resident microflora in newborns can occur through different manners, different ways. So one of the ways is it's passing through the mother's birth canal. The other is receiving the mother's breast milk. So bottle feeding and close contact with people, with air, and also surfaces, they can also expose newborns to different microorganisms. And again, this can contribute to the establishment of the newborn's resident microflora. Contact to disease refers to the progression from the presence of microbes to the development of a disease. Right? So contact occurs when microbes are present, while colonization refers to the presence of bacteria on the body surface without causing disease. Infection occurs when microbes penetrate host defenses, they enter tissue, and then they start to multiply. This is usually followed by the development of a disease, which is the distribution in normal homeostasis or any deviation from health that's caused by microbes or their products. It's also important to note that infection and colonization don't always lead to the development of a disease. So there's two types of pathogens. There's true pathogens, and then there's opportunistic pathogens. Right? So the first one, true pathogens, they cause disease in healthy individuals, and they're associated with a specific and recognizable disease. On the other hand, opportunistic pathogens, they cause disease in immune-compromised hosts and can only gain access to sterile regions of the body, such as the bloodstream or internal organs, when there's an injury or a breach in the body's defense. Right, so opportunistic pathogens, as I said, they don't cause disease unless the conditions are right. Right? So, as I said previously, if there's a breach in the skin, so in other words, we're saying when there's a failure in the host defense system, that's one opportunity. The other is when there's an introduction of the organism into unusual parts of the body. Or it could be when there's a disturbance in the normal microflora. Now, in immunocompromised population, the host's normal defense, they're weakened. Right? And then this allows these pathogens to flourish, to prosper. Similarly, organisms that are normally present in one part of the body, such as your gut, in the case of E. coli, this can cause infection when they're introduced into other parts of the body, such as the urinary tract. So disturbances in normal microflora can also create opportunities for opportunistic pathogens to cause disease. So, for example, taking antibiotics, they can disrupt the normal microflora of the body, and then this leads to overgrowth of yeast, and then it will cause yeast infection. So pathogens are disease-causing agents, okay, and their ability to cause disease is referred to as pathogenicity. Now, the degree of pathogenicity is known as the virulence, which measures the relative ability of a pathogen to cause disease. Now, pathogens, they use various strategies to establish virulence. And opportunistic pathogens will only cause disease when the normal host's resistance is absent. So the virulence of a microbe is determined by its ability to establish itself in a host and cause damage. So virulence factors, these are any characteristics or structures of the microbe that contributes to its ability to do so. So virulence can be estimated through experimental studies of the LD50. Okay, so again, this is lethal dose 50, LD50. And this is the amount of an agent that kills 50% of animals in a test group. Attenuation refers to the decrease or loss of virulence of a microbe. Toxicity is when an organism causes disease by means of a toxin that inhibits the host cell's function, or it will kill the host cell. Toxins are able to travel to sites within the host that are not inhabited by pathogens. And there are different types of toxins that include endotoxins, enterotoxins, as well as exotoxins. Okay, so you got endotoxins, enterotoxins, and exotoxins. Now, invasiveness refers to the ability of a pathogen to grow in a host tissue and inhibits its function without producing a toxin. And many pathogens use various virulence factors that include adherence toxins and invasiveness in order to enhance their pathogenicity. So a pathogen needs to successfully enter the host's body through portals of entry. It needs to attach to the host cell. It needs to evade the host cells, the host's defense system, and then it needs to cause disease, and then it needs to exit the host through portals of exit. Right. So these are all the things that a pathogen needs to, the steps it needs to go through in order for its success. If any one of these steps are interrupted or prevented, the pathogen will not be able to infect the host. Okay? All right, so the portal of entry, what is that? That's a route that the microbe takes to enter, to get to the tissue of that host, right, to start infection. All right, what's exogenous? All right, microbes that originate from a source outside the body, from the environment or another person or an animal. So again, exo means out, right? So exogenous, these are microbes that come from outside the body. What's endogenous? Microbes already existing on or in the body from the normal biota or a previously signed infectious. In other words, endo means within, right? So these are things that are already inside your body. Microbes that you already have. Maybe they're part of your normal, as I say, the normal biota, the normal microbes that we have, that we all have. Or again, it may have been an infection that you had from the past that was silent, that was just kind of hiding. All right, so endogenous, exogenous. Just break down, well, if you break the words down, it's much easier to remember. Endo means within, exo means from outside. All right, so portals of entry. So portals of entry refers to the sites through which these pathogens can enter the body and cause infection. Now, most pathogens, they've evolved to infect specific portals of entry. So if a pathogen enters through the wrong portal, it may not cause infection. So for example, influenza. Influenza enters through nasal mucosa, and then it'll cause infection. But if it contacts the skin, it's not going to cause infection. Okay? Now, some infectious agents, they have the ability to enter the host's body through multiple points of entry. So, for example, Mycobacterium tuberculosis, okay, can enter through both the respiratory system as well as the GI, the gastrointestinal tract. While if you look at Streptococcus and Staphylococcus, they are able to enter through skin, the urogenital tract, as well as the respiratory tract. All right, so the infectious dose is referring to the minimum number of microorganisms that are needed to cause an infection. So, generally speaking, microbes with smaller IDs, infectious doses, they're considered to be more virulent. So, for example, if you look at Rickettsia, just one cell is needed, okay, to cause an infection. While if you look at cholera, it's 1 billion cells. All right. So, and then if you look, everything in between. For tuberculosis, you need 10 cells. So what they're trying to tell you is this: microorganisms that require a smaller infectious dose, they're much viral. They can cause disease much faster, right? So these guys are really nasty, really bad. These guys are not that bad, right? You need to get lots of them before, again, you can start to get sick from them, right? So they're weaker, and these are much stronger. So adhesion is the ability of a microbe to stick to the surface, and it's the first step in causing disease. So in order to initiate an infection, pathogenic bacteria, they must adhere to the host cell through interactions between molecules on the surface of the pathogen and the host cell. So the ability of a pathogen to attach or adhere to the host tissue is important for establishing an infection. So this binding between specific molecules on both the pathogen and the host is what allows the pathogen to gain a stable foothold in the host tissue. So the pathogen is going to be limited to only those cells that to which it's able to bind to. So a firm attachment is almost always necessary for causing disease, since the body has many mechanisms, to remove microbes from tissue. Now, bacterial adherence. Okay, adherence is the ability for a microbe to stick to a surface, and it can be facilitated by extracellular macromolecules like a slime layer or a capsule, as well as by fimbriae and pili, which you find in pathogenic strains of bacteria like E. coli and Neisseria gonorrhea. These structures are also known as adherence factors, okay, or another term that they give for it are adhesins. Okay, adherence factors or adhesins means the same thing. But when microbes enter the host, they're likely to face the immune defenses of the host if they are not established as normal biota. Phagocytes, again, these are cells that eat up or that engulf and destroy pathogens using enzymes and antimicrobial chemicals. So over here you can see a white blood cell eating up a bacteria. So antiphagocytic factors are virulence factors that help pathogens avoid phagocytes. Remember, we said that phagocytes are the cells that destroy the pathogens by using enzymes and other antimicrobial chemicals. So some examples of antiphagocytic factors include leukocidins that will kill the phagocytes, as well as slime capsules that make it difficult for the phagocytes to engulf the pathogens. So we see that in Streptococcus pneumoniae, as well as Salmonella typhi. Or again, for the leukocidins, you'll find them in Streptococcus and Staphylococcus species. Right, virulence factors refers to any substance that's produced by a microbe that's going to enhance its ability to cause disease. And these factors, they may include antibiotic resistance compounds. They could be enzymes that break down the host tissue, or enzymes that interfere with the normal host's defense, as well as it could be toxic substances that are released by the pathogen. So some of the examples, they include neurotoxins, coagulase, and hyaluronidase. But microbes cause damage to their host through 3 ways. either directly through the actions of enzymes, it could be directly through the actions of toxins, both endotoxins as well as exotoxins, or it could be indirectly by inducing the host's defense to respond excessively or inappropriately. So virulence factors, they're adaptations that's used by microbes to establish themselves in a host. All right. Exoenzymes are enzymes that are secreted by microbes, and they can break down and damage host tissue. So these enzymes, they'll often dissolve the host defense barriers, making it easier for the pathogen to spread and cause disease. And an example of an exoenzyme is hyaluronidase, okay, as mentioned in the previous slide. Hyaluronidase will digest the ground substance that cements animal cells together. Okay. Another example is coagulase, which causes the clotting of blood or plasma. Streptokinase is a virulence factor that's produced by Streptococcus pyogenes, which causes disease such as strep throat and scarlet fever. Streptokinase, it's an enzyme. It breaks down fibrin in blood clots, and so therefore it's enhancing the invasiveness of the pathogen. Now, streptokinase, it's also been used as medication to dissolve blood clots. All right, so what's a toxin? A toxin is a chemical product that causes cell damage. Endotoxins and exotoxins are two types of toxins that are produced by pathogenic bacteria. So let's first look at exotoxins. Exotoxins, these can be proteins that are released by bacteria that can cause damage to host cells by disrupting normal cell function. They can be further classified based on their mode of action, such as neurotoxins or enterotoxins, as well as hemotoxins. Endotoxins, on the other hand, they're part of the outer membrane of gram-negative bacteria, and they're released upon cell death, which causes inflammation, or it can cause other immune responses in the host. Now, enterotoxins, okay, they're a type of exotoxin that affects the small intestines. So again, enter means small intestines. What enterotoxins will do is they end up causing massive secretions of fluid into the intestinal lumen, and this leads to symptoms such as vomiting and diarrhea. An example of enterotoxin is cholera, the cholera toxin. Okay, it's produced by the bacterium Vibrio cholera, and again, it gets transmitted through contaminated food or water. So the portal of exit, this is the means by which a pathogen exits its host to infect other cells. Okay, so different portals of exit exist. They include, if you look here, what is this? Respiratory secretions, okay, or could be saliva, could be the skin, okay. It could be feces, right? It could be urine as well, again, body fluids, urogenital secretions, and also it could be blood. So the portal of exit is an important factor in the transmission of infectious disease. So the specific avenue by which pathogens exit is what we refer to it as its portal of exit. Okay. So again, pathogens are able to exit through, as we said in the last slide, secretions, excretions, discharge, or even tissue that falls off, sawed off tissue. The higher the number of microbes in these products, in these materials, the likelihood that the pathogen will reach another host ends up becoming higher as well. So again, the portal of exit is usually the same as a portal of entry, but some pathogens, they use a different route to exit the host. So again, in this slide, it's just going over a look. First thing, how does a microbe survive? What does it need to do? Well, it needs to find a portal of entry, right? And look at all these portals of entry. Now remember what we said. The portal of entry is usually the same as the portal of exit is also going to be probably the same as the portal of entry. So if you look over here, it's almost the same thing that you see over here, right? Nearly the same stuff are found for both the portal of entry as well as for the portal of exit. All right. Aside from that, we also said that in order for bacteria to be to survive, to cause disease, they need to be able to attach themselves, right? So again, these are the things that they use to attach themselves. Surface proteins, they can make capsules, fimbriae, they can have spikes. So these are what the pathogens need. Now, how do they survive? Okay, how do they survive if they are able to attach? Well, they can avoid by not getting eaten by the host's defense systems, right? Avoiding phagocytosis, avoiding death inside the host, right? Or again, when they can be opportunistic, right? During the absence of specific immunity, when the host immune system is weakened, that's how they may survive. And then what are they going to do? They're going to cause damage, right? So that now they're starting to release toxins, right? So direct damage through toxins, or indirect damage by inducing, again, other responses from the host due to their presence. That's the damage. And then after a certain point, they're going to want to go, they want to exit when the conditions are not favorable for them to continue to multiply, divide, and do whatever that they're doing. So now they want to exit and they want to continue into another host. All right, so when we're talking about localized infections, we're referring to an infection that's confined to a specific part of the body, and it has local symptoms such as redness, swelling, and pain. Now, systemic infection, on the other hand, as the name implies, it refers to an infection in which the pathogen has spread throughout the body, and it affects multiple organs, multiple systems. So it's causing a more severe symptoms. Now, a focal infection is an infection that's initially localized in a specific part of the body, but it can spread to other areas. Whereas a mixed infection is an infection that's caused by more than one type of pathogenic microorganism. Okay, so if you look over here, lots of different microorganisms are causing this infection. Now, secondary infections will occur when a new microbe becomes established in the body following an initial infection. Acute infection, on the other hand, is when there's a rapid onset and a short duration, right, of that infection, of that pathogen. Chronic infection is when you have a long duration of that pathogen. And again, a lot of times it's asymptomatic, or again, it may even be a subclinical period. Asymptomatic or subclinical infections, they'll occur when the person is infected with the pathogen, but they don't show any noticeable symptoms. Okay, they're absent. Symptoms are absent. So, we spoke about some terms, signs, symptoms, syndromes, and you guys should all be familiar with this. What is a sign? Anything that you can measure is considered a sign. Okay, anything that's measurable. So your body temperature, right? That's measurable. If you have redness or if you have swelling, that's measurable. Okay, an increase in white blood cell counts, that's measurable. You're vomiting, that's measurable. Okay, now what's symptoms? Symptom is anything that's subjective. Okay, so, my stomach hurts. All right, they say a tummy ache. Or I have pain over here. I feel like I'm dying. These are all subjective things, right? So whatever the patient tells you, what they feel, that's subjective. Whatever you as a healthcare professional is able to observe and measure, those are signs, okay? So objective and subjective. Now, objective things, you can measure them. Subjective things, you can't really measure them. And they're referred to as symptoms. Okay, so objective things that you can measure, those are the signs. Syndromes would be a combination of both signs and symptoms that are occurring together. So again, this is going over stages of infectious disease. So first there's this incubation period. In incubation period, you don't see any signs or symptoms. And then there's the prodromal phase, where you end up having maybe some vague symptoms that occur. Then there's the invasive period. And this is where the severity and the intensity of the symptoms are at its peak, okay, during the invasive period. Then you get to the midpoint, we call that the acme. And then there's the decline phase. And now the number of cells are starting to decrease, okay? And then there's the covalence period. This is meaning that nearly the pathogen is gone. Now you're recovering. Again, over here, they're showing for different type of pathogens, how long is the incubation period. So as you can see, different pathogens, they have different incubation periods. Some of them, they take up to 3 weeks. Others are much shorter. It could be a week, a couple of weeks, as is in the case for rubella, a week to 2 weeks. For a flu, it's less than a week incubation period, right? Less than about 3 days, 4 days for them to incubate. And then, you look at other ones like rabies, for example. Rabies can affect your body for a very long time, right? Yeah, it's like up to a year you may not see it, and then it'll incubate for this period before it starts to show. Same thing for hepatitis. They could be silent for a long time before they they incubate for a very long period. So the persistence of microbes and pathogenic conditions refers to the ability of some microorganisms to remain in the body after the initial infection and to cause disease later on. Okay, and this can happen when the microbe enters a dormant state called latency. Okay, where it's not actively replicating, but it can reactivate and cause disease under the right conditions. Right, so latency is commonly seen in chronic infectious diseases such as those that are caused by viruses, bacteria, protozoa. You'll see it in herpes simplex, tuberculosis, and even malaria. So sequelae. Sequelae refers to long-term or permanent damage that's caused by an infectious disease to different organs or tissue. So for example, meningitis can lead to deafness. Strep throat can cause rheumatic heart disease. Lyme disease can result in arthritis, and polio can lead to paralysis. So what's epidemiology? Epidemiology is the study of how diseases spread in populations. An epidemiologist is looking at the causes, the transmission, the prevention, the treatment, and the number of people that are affected by a disease. Okay, this is what epidemiology is. And this was huge. It got very popular during the recent, the 2019 COVID pandemic. So again, epidemiology, it's a multidisciplinary field that allows scientists to apply knowledge about microbes and the disease they cause to new situations. So it involves the study of disease within populations, and it also investigates factors such as what causes the disease, how is it transmitted, how do you prevent it, how do you treat it, and how many people are affected. So epidemiology involves many disciplines. It includes microbiology, anatomy, physiology, immunology, medicine, psychology, sociology, ecology, and even statistics. It's huge. So again, an epidemiologist, this is a scientist that's studying, that specializes in the field of epidemiology. Now, what are etiologic agents? This is what is causing the disease, right? So the cause of the disease is the agent, the etiologic agent. What's the morbidity? What how much sickness does this disease cause, is the morbidity. Okay, how much sickness is it causing? The mortality it's saying is this disease, this sickness, how many people is it killing? How many organisms is it killing? Okay, that's the mortality. What's the death rate, essentially, due to some disease? Now we're looking at incidence and prevalence. So incidence essentially is the number of new cases, okay? New cases within a specific period of time. That's the incidence. New cases within a specific time period, okay? Now prevalence, on the other hand, it's the total number of cases within a certain period of time. So hopefully that makes sense to you, right? So prevalence from week 1 up to from up to 2 weeks, what's the total number of cases, all right? And you're saying from week 1 to week 2, how many new cases occurred? That would be the incidence. So an endemic can be described as when a pathogen is continuously present in a population, such as, for example, rabies in Morocco. They have an endemic of rabies. Sporadic. So this is when there's when we have occasional cases being reported at irregular intervals and random locations. An epidemic, on the other hand, is an outbreak or higher than normal number of cases of a disease in a population. And again, a pandemic, this is the spread of an epidemic across continents or globally, right? Worldwide. So disease transmission, what is it affected by? Disease transmission may be affected by reservoirs of infection, right? Where is the concentration of this infection? Portals of entry and exit that we spoke about earlier, as well as the mechanisms of transfer. How is it going from one person or one organism to the next? So, to get a good understanding of how to prevent disease, how to prevent infection, you have to figure out how can you interfere with the transmission. So if you can break the transmission at any one of these points, then you can stop the disease from spreading. So again, the reservoir, that's where the pathogen is living. Where is its habitat, right? That's a reservoir. Okay, the source, the person or the item in which the infection is directly acquired from. That's the source. What's the carrier or the asymptomatic carrier? This would be the organism that's harboring that infection that can spread them to others. Okay? And they may show symptoms or they may not show symptoms. So, moving on to vectors, right? So you have biological and mechanical vectors. So biological vector is an organism that carries the pathogen and is also involved in the pathogen's life cycle. Okay? So example they gave is a virus inside of a mosquito or the bacteria inside of a tick. A mechanical vector, on the other hand, they only carry the pathogen and they don't play a role in the pathogen's life cycle. So examples of biological vectors, mosquitoes and ticks, while flies are examples of mechanical vectors. Okay, they're just transporting the pathogen. These guys, biological, not only are they're actually letting it grow inside their body. Okay? They play a role in their life cycle, of the pathogen's life cycle. All right, what's zoonosis? Zoonosis refers to an infectious disease that can be transmitted from animals to humans. Okay, that's zoonosis. So again, COVID is an example of that. Right? It was found in bats. From bats, it went to humans. So other examples, they include rabies as well as Lyme disease. Okay, so approximately 70% of all emerging diseases globally are zoonotic. And eradicating these diseases can be difficult without also eradicating the animal reservoirs. Okay, so efforts have been made to eradicate mosquitoes. as well as certain rodents in order to control the spread of zoonotic diseases. Okay, so the source of an infection can be a human reservoir, right? Remember, we have asymptomatic or symptomatic carriers, and as well as animal reservoirs, right? So animal reservoirs, they could be wild animals, they can even be insects, or they could be non-living reservoirs. Non-living reservoirs, we're talking about soil, water, food that are harboring and transmitting infectious disease, infectious agents. Right? Some diseases are communicable diseases, right? Communicable diseases, we're talking about those that can be spread from person to person or from animal to humans, while non-communicable diseases are not caused by infectious agents, and they cannot be spread from a person to another person. So examples of communicable diseases include what we spoke about earlier: flus, tuberculosis, sexually transmitted infections. And examples of non-communicable diseases, there could be heart disease, obesity, diabetes, as well as cancer. Again, another example of communicable disease, we're saying that it spreads from person to person, right? From host to another. So a flu, a cold, meningitis, these are communicable diseases, right? Now, contagious means that it's easily communicable, right? Contagious means something that's easily communicable. So the flu, it's easily communicable, right? So it's contagious. The same thing goes for measles. It's easily communicable, so it's contagious. All right, now we move on to horizontal and vertical transfer. Horizontal transfer refers to the transfer of genetic material from one individual organism to another of the same generation and typically within the same species. So this can occur through various means, such as direct contact. It could be due to ingestion of contaminated food or water, or even through the air. Now, vertical transmission, on the other hand, or vertical transfer, refers to the transfer of genetic material from one generation to the next. And this could occur through reproduction, where the genetic material is passed from one parent to the offspring. So if you look over here, again, they're telling you that for direct contact, it could be through intimacy, such as kissing. It could be through sneezing. And again, they could be vertical vectors, right? Indirect, we're talking about contaminated material. So indirectly, you're getting it through contaminated food. It could be water, other biological products, like it could be through blood transfusion, through serum, or again, even through fomites, non-living organisms. So like a doorknob, you touch a doorknob, perhaps through toilet seats, or again, it could be through the fecal-oral route. Someone goes to the toilet, they don't perhaps wash their hand very well after they defecate, after they clean themselves, and then they touch their mouth, for example. They can go from, again, fecal to the mouth, fecal-oral route. And then there's air. Okay, so air, we're talking about droplet transmission, right? So it could be dried microscopic residue. It could be aerosol, dust or moisture particles as well. So if you look over here, when a person sneezes, all these particles, okay, so an enormous amount of moist droplets, and these dry droplets, we call them, the droplet nuclei, they can spread. So that's why we tell you, when you want to sneeze, cover your mouth with your elbow, get the inside of your elbow, with the antecubital region of between your forearm and your upper arm. All right, so we have more definitions here. First one, sanitation. Sanitation is the use of measures to promote cleanliness and to prevent the spread of disease. So again, we can do that by proper disposal of waste, having a clean water supply, as well as having good hygienic practices. Positive hygienic practices, I should say. Immunization is the process of becoming immune to disease by receiving a vaccine that stimulates the body's immune system to recognize and to fight pathogens. Moving on to isolation. For isolation, we're talking about separating or the separation of people, separation of individuals that are infected with the contagious disease from those that are not. And we do this to prevent the spread of the disease. So remember, we said from the portal of exit, what happens? It needs to get to another host, right? So if you can stop that, if you break that chain, that the disease is not going to proliferate anymore. Which, again, so isolation goes into quarantine, right? Quarantine, we're talking about the restriction of movement, okay? Restriction of movement or separation of individuals that have not been exposed to contagious disease, but they may not yet be showing symptoms in order to prevent the spread of the disease, right? So isolation, quarantine, right? They go together. Okay, so think of quarantine as a more severe form of isolation. Quarantine, you want to limit their movement, right? Don't leave this room. Stay inside. So what we went through, right? During the pandemic, we were ordered to stay inside, stay in the home for 2 weeks or 3 weeks, however long it was. All right, control vectors. Control vectors, these are measures that are taken to prevent or limit the spread of disease by controlling the population of vectors like mosquitoes and ticks that transmit the disease. Also, most important is education. Okay, education about prevention and treatment. And again, we saw this during the COVID pandemic. A lot of education was being announced all over through social media, through news outlets, through publications, from your doctors, from healthcare providers. So again, education is about providing information as well as resources to the public, to the population, about how to prevent the spread of the disease, as well as how do you recognize and how do you seek proper treatment if they end up becoming infected themselves. All right, and that's all for this lecture. So we'll see you guys next time.